All right, so we're going to start the demo and the lab for diffusion using the agar cubes with phenolphthalein, which makes them bright pink. And we're going to be putting them as a basic cube made with NaOH or sodium hydroxide into a solution of hydrochloric acid, which is a very, very strong acid. So let me screen share as we walk through the general setup. And so we're going to do procedures 1 and 2 in one go. And so you'll have 3 things to take data on, 3 things to do calculations on, and 3 things to graph all at one time instead of doing this across multiple days. Now, the objective is the same. So we're still going to be talking about how cell size and shape affect overall nutrient intake and waste elimination, right? We're talking about diffusion. We're talking about non-active transport, so things just randomly moving around to go inside of a cell or to leave a cell. Okay. Chapter 4 summary and Learn Smart are still due Sunday, so they will be our last grade on this report card. And I'm going to ask you to finish your graphs and calculations of your data based on this data set. So our cubes initially do look like this. I can show you kind of briefly right now. So they're placed here. I don't want to drop them since I just really painstakingly cut them out. And we have our kind of demonstration of what they're going to end up looking like as the white part starts to move inwards because the hydrochloric acid is neutralizing the sodium hydroxide base. And so the phenolphthalein is turning from pink to white. And so the outside is going to start turning white and it's going to start moving in. So lab prep, I'm not going to actually have y'all do this, but you do need to keep in mind that, remember, volume is base times width times height. And then surface area for rectangular prisms is 2 times each of the faces all added together. And so we're going to be finding surface area to volume ratio at the very end to kind of compare the fusion rates. All right, so basically, during lab, we're going to be calculating data for different time periods. And the time periods are actually quite spaced apart. So I'll be recording and then inserting clips when I reach those time points. And I will record those on my phone for timer. And for each data point, you're essentially going to be calculating what I've drawn on the side over here. So don't worry about this part too much. But initially, all of our cubes are going to start out all pink. And they're going to start out, they're going to start out all pink. And after you add them to the hydrochloric acid for a while, they're going to start turning white around the edges. And as the white part moves in, we're still going to want to calculate the amount of pink left. And so that was based on the green-blue cube that I calculated for you guys in the last recording, right? So you're going to be able to calculate the amount of pink based on the distance that you see the white has moved in. So we'll be measuring that in, we'll do centimeters. So we'll be measuring these in, we can do millimeters as well. Okay, so we'll be measuring these in millimeters of how far the white is moving in, right? That is the diffusion amount, the diffused distance that the hydrochloric acid has gotten into the cubes just from sitting there, right? Passive transport, no active transport is occurring because obviously the agar cubes are not alive. And so, for example, if you initially start with 23 millimeters, 20 millimeters and 21 millimeters as your dimensions of your pink agar cube. And after 10 minutes, all of the sides have turned white by 2 millimeters, which means that your base of your pink now is going to be the original 21 minus 2 meters from the left and minus 2 millimeters from the right. And so your new base that you're going to be calculating for your pink is going to be 17 as the bottom, okay? You're going to be doing the same and subtracting 2 millimeters from either side of the height and the width, and then you're going to be calculating the volume of the pink that is left. We also want to calculate volume of diffusion. So that's going to be the amount of white, or the volume of the white part. And that's related to when we calculated the amount of blue left, where you subtracted the volume of the green from the total blue. So you easily have the total pink, right? This total pink is essentially your total white, right? Because the original dimensions now match the dimensions of where the white part is. And you just calculated the volume of the pink. So you're going to be taking this total volume and subtracting the small volume in order to get the volume that has been neutralized by the hydrochloric acid. Okay, so for each time point, we're going to be calculating that. And that's related to the table that you set up for each one. So we're going to be doing one table for the single big cube. We're going to be doing a second table for the big cube, same volume, but cut into 4 little cubes. So those little cubes, we're going to see if their diffusion rate is different because the volume is the same, but there is more surface area. So their surface area to volume ratio is a lot higher. And then lastly, we're going to do a different entire shape. And we're going to be doing a flat rectangular prism, again, that has the same volume or a very close volume to the big original cube. And we're going to be doing a table for 1, 2, and 3, all of those scenarios. Okay, so again, distance of white is going to be from the edge until the pink. What is that distance of diffusion? We're going to be calculating the volume of the pink that's left inside of the prisms, right, inside of our cubes, inside of our cells, in the center. So we're going to be subtracting from the edges, and then we're also going to be calculating the volume that's left around the edges of the white. Okay, so I will probably do the first one with y'all, but starting off, before we even do anything, we have to actually measure our base times width times height of our original cubes for our volume and to find our surface area so that we can actually eventually use those as comparisons. Afterwards, obviously, you're going to be graphing. You're going to be graphing the greatest diffusion depth. So for every time point along the x-axis, you're going to be doing diffusion depth in millimeters, and you're going to be having distance. You're going to graph each distance of white for the big cube, the small cube, and the different shape. You're also going to be graphing the diffusion rate. So as time goes on, you're going to be graphing the volume diffused. So the volume of the white part of the cube, and you're going to be graphing that for the big cube, the small cubes, and the different shape. So this would be the 2 things that you are graphing for. So again, each graph should have essentially three like trend lines that match the big cube, the small cubes, and the different shape. And then lastly, you'll do a conclusion based on using your mathematical evidence from your graph and your calculations to explain a relationship between cell size and diffusion speed. Okay. Afterwards, you'll answer these questions about kinetic energy and how does it differ from potential energy. And then you'll also answer the following questions in your lab report. Okay, so we're going to go back to this page. And so essentially, we're going to need to record our initial data for our big cube, our small cube, and we're going to want to record data for our big cube, our small cube, and our different shape. So dimensions of big cube. So we're going to want to measure that. And before we do that, I'm going to put on some goggles for safety. And I'm going to put on gloves so that my hands don't get acid and base all over them, right? Because that would be dangerous. You have my timer ready. And so initially we are going to go look at so those are the 3 cups that I already set up with acid inside of them, but we don't need those right this second. So we're actually going to be looking at our cubes first of all, and I've already kind of cut them out for you. So we have our big cube, right? It's 1 big cube, 1 big cell. Now over here we have 4 small cubes cut from the exact same mold that the original big cube came from, but we cut it into 4 separate little pieces that are as closely similar to size as possible. And then we have a rectangular prism shape that is a different shape, but as closely similar in volume to the other one as possible. Okay, Notice this agar is quite crumbly and it actually will dye your clothes if it gets onto it. Okay, so easy first things first, let us measure using the millimeters the dimensions of our big cube. And so if we take a look at our big cube, our base, I don't know if you can actually see this. Our base, you can see, is pretty much exactly 30 millimeters. Now that means that our width will be the exact same, which will be 30 millimeters. And then our height is going to be about, let's see, about 27 millimeters. I don't know if that's clear or not. So we have a base of 30, a width of 30, and a height of 27. So we want to record that. So base equals 30 millimeters. So you want to write this down in your notebook. Width equals 30 millimeters and height equals 27 millimeters. So now I want you to calculate your initial volume and your initial surface area for the big cube. That means that automatically our dimensions for our small cubes we can calculate because they are sorry, let me take my gloves off. This is a little bit so our small cubes are our big ones divided into 4. So dimensions of small cubes. So our base is going to be 15 millimeters. Our width is going to be 15 millimeters. And our height is going to remain the same because we didn't cut it into 8 cubes, right? We only cut it into 4 cubes. So our height is going to be 27 millimeters still. And so your volume should be the same because the volume is still the same total of adding all 4 of these cubes up together. But you're going to want to calculate your surface area based on the, you're going to calculate your surface area for one of these small cubes, and then you're going to multiply by 4. Okay, so you're going to be calculating one small cube, then times 4. Okay, and then lastly, we're going to want to calculate all of this information for the different shape. And I'll just give it to you ahead of time since it's close in volume, but obviously it's not exact science since it was cut out of an agar piece, right? So we have 60 millimeters for the base. The width is 40 millimeters. And then the height is 10 millimeters. So it's close in volume, but not super exact. So you're going to want to calculate volume just to show that it's close, but not exact. And then you're going to want to calculate the surface area for this one, right? Because at the end of this whole thing, I'm going to want you to do a surface area to volume ratio. Here, I'm also going to want you to do a surface area to volume ratio. And here as well. And these are not going to change throughout the whole lab. You're just going to calculate these out in the beginning because you need these reference numbers to kind of refer to throughout the lab. Okay, so the first time point is going to be super simple. And your first time point obviously is going to be, change the color so you can see it. Your first time point is going to be 0 minutes. And that's right now, before we put anything into our containers. So for 0 minutes, you're gonna have 3 tables. And so we're gonna calculate the distance of the white at 0 minutes, right? Have we even put it into the hydrochloric acid yet? No. So should there be any white? No. So if there's no white, then the distance of white is going to be 0 millimeters, okay? So our volume of our pink should still be our original volume that we calculated, right? So for example, if we were doing this table for the big cube, the volume of the pink would be 30 millimeters times 30, and then times 27. That would give you 24,300 millimeters cubed. And that would be your initial volume of pink. And your initial volume of white will be 0 millimeters cubed because there is none yet. Okay, so you should calculate these out because you already have them. And then you should be able to, put them into your tables, right? This one would be a table for the big cube, right? Just to make that clear. So you want to make sure you title all of your tables, right? And so we're going to see at another time point coming up what the distance of white is and how that affects our volume of pink calculations. So we're going to go ahead and we're going to put our cubes into our actual acid now. And again, since you are working using this recording, you can pause and go back whenever you need to. If you missed the numbers that I put up before, or if you missed the calculations that I was explaining, you can always pause and go back in there. So we're going to have one where we put our big cube, one where we put our 4 small cubes, and then we're going to have one where we attempt to fit our large cube and submerge the whole thing, which means that I need to add a little bit more. And so if you are taking a peek, you can already see that the edges of these are turning a little bit white. And that's because naturally diffusion is already kind of happening. And I'm going to go add a little bit more water into this one. Just so that our concentration stays the same. That's not the thing that's affecting diffusion rate. And I want to start my timer. As you can see, it's starting to become really, really obvious that the white part on the outside is where the hydrochloric acid is neutralizing the NaOH. So we'll see in about, let's take our first time point around the 10-minute mark and see how it's going. So if you are going to wait, I will include time points inside of the post to tell you at what points we actually collect the data for the distance of the white diffusion. Actually, I think I can just record separate ones. Yeah, I'll just record separate videos at the different time points, and we'll take the data at each time point.